real thrill for us to have you here. First of all, tell us a little bit about your laboratory and the scope of the work that you perform.
Well, my laboratory has been in existence for about 40 years now, so this is a very long arc that we can summarize pretty briefly. I've been interested in retinal wiring, how the retina puts itself together, how the retina is wired in the normal retina, how it processes information. And my colleagues and I knew that we wanted to do this as far back as the early 70s, but the technology wasn't available. In fact, we really weren't able to begin on this sort of project until 2009, when the price of a terabyte dropped to the level where an ordinary researcher could afford large-scale data sets. And we started aggressively at that time, and we've been very successful in building large computational models of how the retina really works. In parallel with that, we spent a lot of time developing molecular markers that would allow us to visualize different classes of cells in the retina in conjunction with all of these networks. And then while we were doing all of that, colleagues dragged us rather unwillingly into the world of retinal degenerations. We first started with retinitis pigmentosa models, with human tissue, and then animal models, and then of late we've been working with macular degeneration. And so all of these tools, these computational tools for building networks, these molecular tools for tracing cells, got applied to these retinal diseases, and we found some things that we did not anticipate.
Tell us about that.
Well, specifically, the idea in retinal degenerations is that photoreceptors are somehow compromised by either gene defects or environmental defects, and the photoreceptors eventually die, leaving people without light input to their retinas. And for a long time, the field just stayed with that idea that the photoreceptors had been lost and that the neural retina remained behind essentially unchanged. The paradox was, since I was not trained in these diseases, I'd come from the world of central nervous system organization and diseases, and our concepts there were that when you cut off the input to a system that we call deafferentation, that system remodels in response to that loss of input. And it became very clear from the tissue samples that we received from our colleagues associated with the Foundation Fighting Blindness that in retinitis pigmentosa that the human retinas were severely remodeled, and we didn't understand how that process had become initiated, really what it was. And then one of my younger colleagues, Dr. Brian Jones, discovered that this remodeling happened in all of the animal models that we'd been working with. So it was general, and it fit the model of brain reorganization in diseases like epilepsies. This was relatively controversial for a while in the ophthalmology field, but now is pretty well accepted as the fate of the retina once you take the photoreceptors away. So it's not, retinal degeneration turn out to be much more complicated diseases than just the loss of the photoreceptors. But our latest work, which really was a consequence of a collaboration with a group in Japan that developed a large animal model, a rabbit model of retinitis pigmentosa that lives a long time, in fact lives 3 times longer than a mouse. We were able to follow longitudinally what happened to the retina, and we had casually expected that the remodeling would get bad and then it would really reach some sort of stasis and the retina would remain remodeled. What we found is that the retina continues to degenerate. So this is really a very dark outcome, that the cells continue to die, and at the end the retina is decimated. Neurons are gone, networks are gone, and the remaining cells are compromised in their metabolic function. So it really looked, to us, it looked more like CNS neurodegeneration or even cancer than just a retina that was left behind. And in exploring that in great detail, we discovered that there were mechanisms in play that are exactly like the mechanisms that operate in Alzheimer's disease and Parkinson's disease and a whole range of neurodegenerations, that the same molecular culprits were present in the eye, and that they appear to be malfunctioning just like they do in these CNS diseases. At one stage, this looks to be a horrific sequence of loss of photoreceptors, of remodeling like epilepsy, and then ultimately going into neurodegeneration like Alzheimer's, and that's all very frightening. But what has happened is we have harmonized these retinal diseases with well-studied brain diseases, and so now we're not alone in the dark laboring away. We have a vast body of knowledge now to call upon to begin to explore these in more detail, and we may have been able to flip the story around in the sense that these brain diseases are very hard to study. They may be much easier to study in the retina. And what we said initially was, if we can't cure Alzheimer's, we can't cure the neurodegeneration. If we can't cure epilepsy, we can't cure the remodeling. But the flip may be true. We may be more readily able to find interventions that deal with the remodeling, that deal with the neurodegeneration, which would be a great success for everyone in retina and brain simultaneously. The other thing that it says is that all of the work that is being done, especially here, as described in this AMD conference, to determine what the risk factors are up front and what are the molecular mechanisms causing photoreceptor death, it makes all of that much more important. It's a much more intense effort because we want to prevent retinas from getting to the remodeling stage, from getting to the neurodegeneration stage. Unfortunately, many of our patients are already in those stages. And so we have to aggressively find ways to try and roll back these events. I am not saying that it's possible. I do not know that. I do not know that.
Are there any markers that you're looking at for early detection and then progression?
Yes, absolutely. Well, we know in remodeling that one of those markers is a pathway that's well known in developmental biology, retinoic acid. Retinoic acid's involved in maintaining your skin, it's involved in growing dendrites on neurons in the brain, it's involved in growing the retina in development in the first place. And this is a very complicated pathway that turns on more than 350 separate genes when you activate it. But one of the things we know that it does is it triggers, in part, some of the retinal remodeling. So that's a molecule for which there are some intercessions based on a long history of anti-neoplastic drugs. So we can steal from cancer and learn how to control this part. So there is definitely hope on that front. But it looks to us as though the remodeling and the neurodegeneration are, in fact, parallel arcs, not a serial process. And so we will have to deal with both of them. And the problem with the neurodegeneration is that it's slow and that the molecular partners are very much in debate, even though we've found some of those markers that indicate that cells are under what's called proteinopathy stress. It's too early to say that the markers we've found are the right ones to target, but we've been able to find them very, very early as remodeling starts. So it suggests that the event, to us it suggests the event that the photoreceptor death itself is almost like a grenade going off in the eye, very much like a traumatic brain injury, and that this neurodegeneration is a consequence of that photoreceptor death event. If there are ways that we can ameliorate that death event, we may not be able to stop photoreceptor death in inherited degenerations. We hope to stop it in AMD, but what we might be able to do is keep the downstream consequences minimal. And if we can do that, then all of the new technologies, optogenetics, photoswitches, programmed stem cells, and our prosthetic retinal chips, all of them are potentially enhanced in their outcomes by preserving the retina better. The processes that we are seeing are in fact pretty much independent of the immune system, although you will see inflammation being part of the process that drives the neurodegeneration. But the inflammatory process itself is in fact not the real culprit here. The real culprit is this aggregation of unusual proteins. So there are about half a dozen in the brain that have the tendency to clump together and choke cells up. And we have found one major protein that is also, I'll say the name, it's called alpha-synuclein, and it's common to Parkinson's disease, multisystem atrophy, often found in Alzheimer's disease, and found in a number of other dementias like Lewy body disease as well. So it's a very common protein. It's present in high concentrations in lots of brain cells, but it's very much like mad cow disease and diseases like Creutzfeldt-Jakob disease, that once this protein misfolds and starts to make aggregates, it starts to induce other proteins of the same kind to make bigger aggregates. And it seems like small versions of these molecules, small aggregates, might even be more toxic than the big versions. And it's very hard for us to track them. But it's a very strong signal, and it suggests that the neurons in the remaining retina and retinal degenerations are always at risk from their own biology after this tremendously traumatic event.
So you're saying these proteins act like prions?
Well, so I will say generically yes.
Right.
And Stanley Prusiner, who won the Nobel Prize for working in this area, has now gone on to work on alpha-synucleins and their behaviors, and he would say that. He would say that as well. And there are other workers who say it's not exactly quite like that. It's not the induction of normal alpha-synuclein into a toxic state. It's in fact, by another toxic molecule, it's in fact that alpha-synuclein kind of wants to do this anyway as part of its normal trafficking. And so the question is, why do the aggregates so big? So there's a lot of debate about the chemical details,
right,
but the behavior is of a prion.
So when we look at that behavior, what are the potential therapeutic interventions that might make a difference?
Well, the one that's being talked about the most right now in the CNS is immunomodulation, finding the right kinds of antibodies that will block the aggregation of these proteins. And there have been tests in animal models that have been successful. There have been tests in animal models that have been very unsuccessful, and as has been typical of all of these fields, moving it to human patients hasn't really worked out very well yet. But I think this is actually the right track, and it may not turn out to be immunotherapy that's the right strategy. There are a whole new range of approaches to work with living cells, click chemistries, where we can actually do very specific modifications of specific groups in living cells. The real problem is delivery to patients, delivery to animal models. But I think the hope for intervention is very, very bright. The work will be very, very hard.
So tell us about your next steps in this particular line of research. What is next on the menu for your laboratory?
Macular degeneration. Well, part of what helps us in what we do is developing very robust animal models. And the problem with the animal models is that they very often manifest diseases in ways that are different from what we would desire as a scientist. What I'd really like to do is turn on retinitis pigmentosa in a little tiny patch of cells in a normal retina and follow those cells to learn what the spatial rules of these diseases are, because there's a lot about the physics and patterning in these diseases that is quite obvious on the clinical side and very occult to us on the research side. We can't replicate that geographic variation. AMD is a natural experiment for us that we didn't realize would be so powerful. We've discovered that the remodeling begins right underneath a single drusen, and it doesn't spread outside from that. It's stuck to right beneath the drusen. So it says that the chemical signals, the remodeling signals, are very, very constrained to specific areas. This gives us a real strong advantage in trying to define what the molecular signals are and where we can intercede. It gives us a better control. And these types of models are being developed in mouse right now, but it turns out that large libraries of human AMD tissue are becoming available, and we've been working very hard on the other side developing brand new molecular marker technologies that can mine them more intensively and give us snapshots of what's happening in the neural retina. So I have a bunch of young people in the laboratory, and I expect to see the sweat on their brows.
So finally, Professor, tell us what the implications for patients would be. If you were to be addressing patients, and tell them what the implications might be 5 or 10 years hence of your particular line of investigation.
Sure. The very first thing that, that triggers a little bit of fear in me, because of what we've done is say that these diseases are harder and they're more complex. But what I say about that is not knowing the truth is no benefit at all. And so I think what we have discovered provides a background where we can say, now we need to know how these processes can be modulated, how they can be controlled, so that we can use this parallel stream of pretty amazing tools that have come along in terms of optogenetics, photoswitches, programmed stem cells. I will say that although I'm a very strong supporter of the chip as is being applied right now, I think that the optogenetics and photoswitches hold the real future of being able to rescue retinas in ways that we didn't anticipate. But we will have to stop these other processes. And one thing I'll say to the patients that I wish also to say to the physicians: clinicians and scientists who work in the central nervous system don't pretend that epilepsy will be reversed. They don't pretend that neurodegenerations will be reversed. We shouldn't pretend that the retina somehow has a magical way to reverse all these processes and go back to the way it was. I will say that it cannot do that. The physics are against it. The biology is against it. There may be some magical process by which that happens. I've seen no evidence that it exists. So that makes it necessary for us to intercede aggressively. And under those circumstances, and this is a little bit controversial, I will say that these new intercessions probably need to be tried earlier in disease rather than later. And right now we wait till people are really at the end stages of disease to try these things, and that's probably the worst state to try them in. So I think the future looks good now that we know what we're dealing with as a complete set of diseases, and the challenges are very clear. The question is, are we up to those challenges?
Thank you, Professor. That's beautifully put, very eloquently put.
Thank you.
I appreciate your time, and enjoyed